moving on to our last presentation of this group, and that is with Avalo.ai. And I'd like to bring up Alex Copeland from IndieBio to briefly introduce the founder.
Cool. Hey, Nick. Hey, everyone. Thanks for having me. I'll be short and sweet to not take too much of Mariano's time, but Avalo is in our current 11th cohort at IndieBio, which started in mid-February. We've been chatting with him and Brendan, his co-founder, for I think at least 6 months prior to that. And I'll say, without spoiling anything, that I think these guys have really impressed us, I think, with their creativity as well as their kind of ability to take feedback and iterate. They came in, last year to conversations with a really powerful platform, kind of this hammer looking for what is the nail to be focused on. And I think they've built that technology and that platform up a lot, as well as really found their stride in terms of kind of directing it at kind of some very high leverage and important problems in the world. So, they still have some more time before IndieBio, and I think I'm really excited to see how they keep evolving, and hope you guys enjoy hearing from them. I think that's all.
Thanks.
Thanks so much for the intro, Alex. Really appreciate it. And thanks so much to the organizers for putting this on. This has been a super fun session. I'm really glad that I've been in here. One thing I will say is I think we're a little bit earlier on in the ag process than some of the other talks, so you'll have to just bear with me. So yeah, I'm excited to talk a little bit about what we're working on at Avalo.ai. We're building the future of plants using the next generation of gene discovery. So before I dig in, I just want to mention a little bit about who we are. So I'm Mariano. I'm an evolutionary biologist and a plant biologist and a computational geneticist. My co-founder is Brendan Collins. He's a cell biologist and software development specialist. And I just want to give you a quick overview of our company kind of by the numbers before I dig in. So we are a gene discovery platform, and in the last 2 weeks, we've analyzed 16 traits. We've developed 5 collaborations. We found 32 new gene targets for cold tolerance in rice. 1 week, 2 weeks ago, we parsed 10 million nucleotides in a week. Since we started IndieBio, we've been able to realize a 12-time speed up in our processing. And over the past 2 years of academic and corporate development, we've done over 500 validation simulations. We now work in 6 species simultaneously, so I'm really proud of how far we've come. So the central thesis of our company is basically this, that the future of food depends really critically on developing new varietals of plants. So perhaps some of you guys have seen this figure before. Everything in red is bad, spoiler alert. So everything in red is experiencing a negative change in yield between now and 2050. And of course, that doesn't necessarily mean that the corn kernels are getting smaller. But in fact, what's happening is that extreme weather events are changing the ability of plants to thrive in agricultural regions. Seawater intrusion is reducing the amount of available arable land, pushing people farther inland, and economic demands are reducing the number of people who are actually available to participate in the agricultural economy. And so what we really need are a new set of plants for the 21st century that are able to thrive in this new environment and are able to feed into the processes that we think are really important for the next generation of agriculture. So when I imagine a future of crops, I imagine cold tolerant rice that can be grown by people who are moving farther inland, higher in elevation, who need to continue their livelihood. I imagine rust resistant wheat that is able to grow despite increasing fungal outbreaks from heat waves. I imagine alternative sources of rubber that don't rely on a deforestation model that can supply the increase in demands over the next 20 years. But I also imagine high performance organisms for biological fermentation that can create these fantastic new products in an incredibly small amount of space. And at Avalo, we're actually working on all of those projects right now in cooperation with our partners. So we have projects in rice right now, rust-resistant wheat, rubber-producing dandelions, and microbial projects. And the way we do that is by understanding biology as it is. So a traditional model of understanding the biology of plants has really relied on simplifying the relationship between genome and trait. But as scientists and as farmers, I think we know that that's not really the case. So the genome actually interacts with itself, with other genes, with the epigenome and the transcriptome to create proteins. And those proteins actually generate the traits that we're interested in. And that trait doesn't just exist in a vacuum, in a growth chamber in a university. It exists in a dynamic environment with humidity and light and heat, and a thriving microbiome that interacts, that sends signals to that plant to change plant outcomes. At Avalo, we've developed a model that can incorporate all of those inputs and find targets despite that. But existing models weren't built to do that. And I think that's pretty clearly evidenced in the genome-wide association study, which has been our main mechanism for finding gene targets of interest for agriculture for the past 2 decades. Every time we turn one of these analyses on, we generate hundreds of false positives per run. Each of those false positives, on average, is worth $40,000 in wasted time, wasted effort, wasted equipment. And that brings the total cost of discovery up to $20 million. And this explains why breeding tools have really only been available for major row crops. But we know that we need to democratize the breeding system to be able to domesticate new types of crops, like the aquatic plants we heard about earlier in this session. And so what we've developed is a new mathematical technology at Duke University that we are calling gene discovery via informationless perturbations. I won't go into the science behind it, but what it provides us is an evolution-aware noise reduction that allows us to find more true positive targets without bringing in those expensive false positive targets for breeding. And that lets us lower the overall cost of a breeding program tremendously. Our software is cloud native, which means that we can scale up to as many partners as we can accommodate. And our process is data agnostic. And being data agnostic, I think, is actually pretty cool because it means we can do discovery in any market. It's really exciting, actually, to be in the world of genetics right now in agriculture because there's this whole biology revolution happening in the agricultural sector. Of course, CRISPR and plasmid deliveries have gotten a lot of attention lately, and mRNA from the vaccines that some of us have, and I hope we can all have soon. But also, people are working to change soil microbiomes to change plant traits, and that can benefit also from our microbial discovery process. And even traditional breeding actually relies pretty critically on gene discovery. So molecular breeding has been used for a long time using genes to stand in for traits of interest. And now, with the advent of big genomic data, we can actually engage in predictive breeding, which maximizes genetic gain by predicting which 2 plants to cross that will result in the maximum genetic gain. We're pairing our high throughput computation with high throughput biology. So we can actually match the speed of our algorithm, the speed of our lab work. So to do that, we're bringing in advanced molecular biology techniques like CRISPR-X, lipid nanoparticles, and RNAi to minimize the space between the theory and the computation and the actual practice. And that lets us quickly translate those into predictions, again, not just for traditional breeding processes, but for synthetic biology. So for a business model, we're really thinking about models that are similar to, let's say, Atomwise, which is working in a different sector, or other computational companies that have worked in the biology sector. And our basic thesis is that every project we take on, every data set that we bring in has value, because every data set that we show to our algorithm makes our algorithm smarter and makes discovery easier in the future. As we participate more in the validation process, as we use our molecular biology tools, we capture an increasing amount of that revenue. And so we shift from a revenue share that's something like a 20-80 split in our favor to something like an 80-20 split in our favor. And over the long term, we're really interested in bringing the entire development and prototyping chain in-house so that we can really capitalize on all of the improvements that we can generate in the breeding process. And since we started IndieBio, we've been laser focused on developing this business strategy. So in the beginning, we were really focused on validating our algorithm, showing that we were able to really develop this big improvement to the breeding process. Now, what we're really focusing on is turning the collaborations that we built into revenue sharing contracts. We're also working to bring the synthetic biology in-house to, again, minimize the time between computation and validation. And of course, we have ongoing field trials in Argentina, Japan, and hopefully soon the US to generate our own proprietary data and really showcase how fast we can make improvements even out in the field, even in a complex environment. So we are raising a seed round in the next couple months here, shooting for the beginning of July. And for our seed round, what we're really focused on is scaling up our processes. So we want to bring in at least 100 high quality collaborations with high quality data sets. We're looking to convert about 30 of those into royalty sharing contracts. Of course, all of them have value, but we'd like to actually take a share of the resulting revenue from the lines that we help develop, and that provides lifetime value to our company. For the most promising of those projects, we want to bring 3 fully in-house and actually develop new lines of plants to sell on our own. And I would like to finish by just talking a little bit about our amazing advisory team. Cynthia and Kathleen, both at Duke, participated really heavily in the development of this algorithm. Daniel has been our mentor in understanding the relationship between genomics and agriculture and business strategy. And Landon has been our superstar, helping to scale up our platform to terabytes of data. And of course, thank you to the IndieBio team. We absolutely 100% would not be here without them. So thank you so much for listening. And I'm happy to take questions about anything you were interested in.
Great, Mariano. Thank you so much. That was very enlightening, and you know, the scale and speed that you are able to work on, considering you're just raising your seed, I think it's very impressive. You've done a lot of work. Any questions right now? Feel free to hop in if you do. Sure, we have some out there. This audience. Go right ahead, anyone? If not, I'm going to put Alex on the spot. I'm sure he wouldn't mind, right?
Alex gets to ask us questions all the time.
You asked Mariano and Brendan a lot of questions.
Well, Alex, how about you kick it off for us? When you first heard this, and as this idea came to you, what questions are in your mind? How do you look at this industry?
I think one of the big questions we had is, how does a company like Avalo break into what is kind of dominated by a few major players? And so for us, we've looked a lot at how do you build collaborations, and I think that's because I think Mariano touched on quite a bit, but one of the key things here is building, I think, a really strong pipeline and understanding the market beyond just the big 3 ag players. So I know, Mariano, maybe it's an interesting thing to talk about is maybe a little bit more on all your collaborations and relationships and kind of who they're with and what you guys are kind of learning from them.
Yeah, happy to talk about it. So we have been really focused on, we really want to demonstrate our value, and we really want to do it in a collaborative way. And one thing we found really early on is that in agriculture, information about plant performance and the genetics of plants tends to be really siloed, and it tends to be really siloed in some very big actors in the field. And that, for us, for a computational and breeding platform, wasn't conducive to building a lot of data and making a lot of progress, particularly in the kind of specialty crops that we've been interested in for kind of a while. So we kind of went out into the world and just started talking to people, and we've developed some really amazing collaborations. At NC State, for example, we're working on rust resistance in wheat, in hexaploid wheat, which has particularly been challenging because of the polyploidy. We've been working in pine. There is more of a predictive breeding process. With Ohio State, we have been working on the rubber producing dandelions. It's a new crop. We're really interested in being able to scale up and grow with a new crop on the market, which is really exciting for us. But we've also been really interested in being able to showcase the other applications where genetics can make a really big improvement. I think a lot of people think about genetics and they think about, okay, we're going to increase yield. We're just going to make like grain size bigger or something. But we can participate in other processes. Genetics has a role to play in increasing industrialization by making plants more uniform and easy to harvest mechanically. It plays a role in industrial processes by making compounds that are easier to extract. And it plays a role in microbiology, which has become, I think, is this really exciting part of agriculture right now. So, to that end, we have started to work on gut microbes in cattle, which, cattle are not plants, microbes are not plants, but our process we found is really flexible and can work there as well.
Great. And I saw Rob Trice put a question into the chat bar about your competitors.
Yeah, I mean, I'm happy to answer. Yeah. So our competitors, we really see that there's a lot of excitement around plant breeding right now. Benson Hill, of course, has been in the news recently. Inari is a really big, really well-funded competitor. One advantage we think that we have is that because we approach this problem in a fundamentally different way, we have much lower data requirements for entry. We don't need 2 decades of understanding about the performance of hybrid corn in the field. We can actually work with very little data. And one thing that's been really exciting to us is that lets us work in spaces where some of these very large competitors can't necessarily work. So we've had a lot of really interesting conversations around tomatoes, which is a really exciting specialty crop. It doesn't have the kind of long history of breeding like a row of sort of hyper-intensive breeding like a row crop, but nevertheless has some existing data that we can leverage. And we need very little just to get started. So yeah, we've had a lot of traction, I think, in some of these more niche markets that a lot of our larger competitors are sort of have some challenges working in.